We are recording this session with permission from all of today's speakers. So without further ado, I would like to introduce our first speaker. This is Dr. Fred Schumacher. He is Associate Professor of Population and Quantitative Health Sciences and co-lead of the Cancer Prevention, Control, and Population Research Program at the Case Comprehensive Cancer Center. Fred?
Great, thank you. Hopefully everyone can hear me. Okay, so everyone should be seeing the first title slide. So I'm going to talk about, first of all, thanks to the organizers, Dana and Jessica, for the invitation. And thanks for everyone who's joining, taking your time out of your day to hear this. And I'm going to present the genetic and polygenic risk scores. And I'm going to look at a primer with prostate cancer. So first is like kind of defining what we mean by polygenic risk scores. Sometimes these are referred to as the genetic risk scores or the PGSs, same thing as the polygenic risk scores. And it's really the idea of capturing an individual's genetic liability to develop a trait or a disease. And it's really thinking about how to quantitatively measure that in a cumulative sense. Not much different than from when you would screen populations for Mendelian risk, where you're just looking at one or maybe two loci. And if someone is a carrier, then there's a high probability, or their genetic liability for developing that disease is much higher. What we'd seen with things like GWASs and some sequencing studies was that the effects that we were capturing was much smaller. So this was then putting them in more of a quantitative cumulative measure. And really this was kind of first done, at least from what I remembered, was around 2009 when the International Schizophrenia Consortium group looked at a polygenic risk score. They defined it then just as the polygenic variation and how it contributed and captured schizophrenia and bipolar disorder. And at this point in time, they looked at the top, I want to say around 20,000 loci that was associated with schizophrenia and bipolar independently. And they used a threshold of around a 0.05 p-value. Don't quote me on that. It could have been a little slightly different. And then LB pruned those variants, came up with a score and saw how much of the variation was being captured. And then looked at, if you cross-compared the schizophrenic score to bipolar disorder, how much did it capture of that phenotype? And you would expect a little bit higher compared to other phenotypes like cancer, and I think they included cardiovascular disease in there. And the idea being that this was demonstrating that it was working, and then that they could also look at what some of that shared variation would be between two different or two similarly related traits. And then within like a year's time, Peter Vischer's group, led by Jian Yang, also looked at this for a quantitative trait and looked at how much of the common variation captured heritability of human height. So just to think about some of the applications of what the polygenic risk scores, I think the first things people sometimes think is because it's individual, that you can get it on each person, that we're looking at some sort of prediction modeling. And that's definitely true to some level when you think about personalized medicine or the clinical impact and how you could use and incorporate these scores into like maybe even a screening paradigm. It makes sense to think about it from prediction. But there's also a lot of other areas that the PRS could be applied to. And one was, as I just pointed out with the schizophrenia and the bipolar, was that as how much does the shared genetic etiology exist between two traits. And this can be important to kind of understand how you might want to group certain diseases. You could think about it from the whole public health spectrum of like, if you have two traits that are very similar, if you have a screening program for one, maybe that's a way to kind of capture the other ones. You got the primary prevention and the secondary prevention possibly on drug treatments, and then how you could care for that. And there's plenty of examples in the literatures and others presented here where we had schizophrenia and depression, but then in the cancer world, people have been doing this on several different levels. Particularly a nice paper by Sarah Lindstrom looked at around 14 different cancers and how they were genetically similar to each other. And this has also been applied a little bit easier in something like LD score regression. But the difference with LD score regression is you don't get the individual score. You just are looking at the summary across the traits. The other areas to think about for the applications of PRS is in maybe a design or a hypothesis test. So for one thing, one area that we're looking at within prostate cancer is trying to find environmental factors, which have been very elusive to this state so far, by stratifying on the polygenic risk score, right? So we could look at it by the top quartile or top quintile versus the lower, and seeing if there is any differences by a environmental interaction. And then there's other individuals who've done this through Mendelian randomization as well as just stratification. So how do you approach to develop a polygenic risk score? So the first step here is just to have a discovery GWAS. Now this doesn't mean that you have the discovery GWAS from working with a mentor or it's in your own lab. It's probably most likely going to be a publicly available database, because here's where you don't need the individual data. You're just capturing some of the summary statistics. So you can see if someone's done a large GWAS and they found multiple signals across the chromosome, across the genomes, then what you can do is take these summary statistics and try to capture those SNPs in a target sample. So that target sample is going to be individuals that have overlap with the same genotype information. So that's going to be key. So obviously, if you're going to quantify the genetic signals from the discovery GWAS, you want to make sure those same signals exist within your target sample. You want to make sure that the QC is up to par, where you have the same strand, the same alleles. And then you may want to consider some of the differences in LD. Now, it doesn't necessarily mean the target sample has to be the same ancestral group or even necessarily the same disease, or it could even be a subtype of the disease. But that's up to the individual investigators and what they're trying to actually do. If you're trying to validate your signature to begin with, then you're probably going to want something very similar to what the discovery GWAS was. And that's the way I'm going to portray it and describe for prostate cancer. So the next step is to come up with, using the betas or the effect estimates from the discovery GWAS, how do you come up with a profile within your target sample? And this is just a very simple example of looking at 5 polymorphisms. You can see those listed from here, from left to right. And down at the bottom, you're going to see that this is the beta that came from our discovery GWAS for each of the alleles, or for one of the alleles at each of the loci. So here we just looked at loci 1. The beta for the C allele is in a protective effect of minus 0.02. And you can see that the other ones, how they correspond going across those loci. So within our target sample, where we have the genotypes on the individuals, we're going to take that beta and we're going to multiply that by the number of alleles for that individual at that locus. Okay, so for the C allele, there's only one representative seal, so we're only going to include the 1 times the negative 0.02. Where in the next locus, there's 2 G alleles, and it's going to be multiplied by 0.01. And you're going to sum those up across, and you're going to get the polygenic score. And from there, you're going to want to then evaluate that in your target sample, usually looking at some sort of distribution. So whether you're looking at the top decile or the top percentile or quartile, however it is, you're trying to find people who might have the highest risk, being those that have the highest polygenic score, versus individuals somewhere down in the lower part of that spectrum. So that's just a brief overview. So now I want to move to prostate cancer and kind of show what we've kind of done and some things that we're still thinking about and how to develop this. So first, just a very quick overview of prostate cancer for those that aren't that familiar with it. It's a really good cancer to look at for when you're thinking about the heritability of cancers. It's got one of the highest familial heritabilities where you're looking at, the narrow sense heritabilities from twin studies. It's over 50%. I think it's second only to melanoma. It has very few environmental risk factors that are consensus across all groups. And the reason why that's kind of nice is because then you don't have to worry about some of the messiness of collecting some of that information from some of your validation studies. You can just really worry about several things, one of them being age. So it's definitely a disease that increases as you age. It also has a direct effect across different continental ancestral groups. So if you look almost like a stepwise distribution across from both incidence and mortality, where Asians are at the lowest risk for both, and then intermediate would be the non-Hispanics, whites, and then African Americans, and then Hispanics, depending on the admixture, would be between some of the Asians and the Europeans or the whites. And the other thing is it's got a high familial history of prostate cancer. The risk increases as the number of individuals that are infected. So where were we with this back in 2017? Prostate cancer, most people have probably seen something, a similar type figure where you have the risk effect plotted here on the y-axis, where it's the lowest risk down toward the origin and increases up to the greatest risk. And then that's being compared across the different allele frequencies. from the lower or more rare polymorphisms or variants to the more common ones. And yes, prostate cancer has a few rare variants that we do know about. The effects are a little bit smaller than what you've seen for, like, say, breast cancer or ovarian cancer, where BRCAs are only about 3 to 4-fold risk, unlike like the 6 to 10-fold within breast cancer. And then there's a few other ones that we've seen from CHEK2 to ATM, and then even from a GWAS, there's been a recently discovered one, HOXB13. But the majority of what we know is coming from the common variation. And you can see the ones that are listed here. And currently at the time, there was about 100 prior to 2017 markers that's been associated with prostate cancer at the genome-wide association level of p-value of 5 × 10⁻⁸. And now we're up to over about 170. And of course, most of these have very similar features to a lot of the traits. They're more common, so typically above the 5% allele frequency, and usually the effective range is from 1.05 to 1.2, so very low effects. So our discovery, GWAS, luckily was one that I didn't just have to go deal with and get the summary statistics. This is one that we were actively involved in. So this is something that was published in Nature Genetics in 2018, and this is where the largest prostate cancer meta-analysis that was available at this point in time. And you can see here, we didn't have, like, fortunately, unlike other traits, we didn't have an issue with numbers. We had over 82,000 prostate cancer cases, another 61,000 controls. And then nicely for looking at different kind of strata, we also had 15,000 what we defined as aggressive cases at that time. And you can see the number of SNPs did vary from up to 1 million down to 43,000. So obviously some of these studies contributed a little bit differently to the overall meta-analysis, but you can see that we still had a very large sample size. And even though at that time we had 100 known variants, we were still able, by adding this last piece here, this OncoArray study of 46,000 cases and 27,000 controls, we were still able to discover another 65 new and novel variants. So one of the first things we did, and this is kind of common in cancer, and we can discuss this later on in the Q&A part, is we looked right away at what is the risk by the percentiles. And so we looked at it from a couple different percentiles, from the two extremes of whether you're a very high carrier. So if you remember that distribution that we showed earlier, this would be the individuals over here to the 99% versus the ones that are less than 1%. The other thing that I think isn't always common across polygenic risk scores is who's the comparative group. So a lot of people will like to compare the two extremes of the 1% or maybe the lower decile to the upper decile. And then you can get very large effect estimates. We like to tend to think that we want to compare it to the average within the population. So that's why we use the interquartile range the majority of the time. And this gives us a better idea of where our risk assessments are for those extremes. And you can see that we're, in the upper 99% for this study, it almost reached 6-fold, versus when you're down to 1%, it was an 85% decrease. And the confidence intervals here were significant. Now, since we used our own study in the betas, this is slightly a biased sample when it comes to the p-value and the significance. The effect estimates we showed in that paper that demonstrated that it's very much the same. And as your sample size increases, and other people have shown this from Naomi Ray's groups and others, that as the sample sizes increases, you're going to have a less bias away from that effect estimate. And then just looking across the deciles here, we wanted to just show and look at different substrata. So remember, going back to the polygenic risk scores, this might be something for future development of studies of how we would want to focus particular groups, or maybe where the most optimal screening could be implemented. And really, we don't see much difference from the overall versus the aggressive prostate cancer. We didn't see much difference from family history, but we did see a little bit, or a significantly increased risk estimates for the under 55 years of age. And this is something that we're still working on and trying to follow up. Luckily, sometimes you get lucky where a study who was supposed to be part of the OncoArray group or the consortium at that point was unable to get their genotyping complete. So because of that, they weren't in the initial paper. But so then we've been looking at them as a independent validation. And this is a study out of France called the EPICAP study. Its tendency is a little bit small. So it's only around 679 prostate cancer cases and 700 controls. But we were able to at least look at the majority of the SNPs that came through the quality control protocols, showed what the distribution looked like here. For the orange colors are those in controls, And for the green colors, these are the prostate cancer cases. So you can see that the distribution of the risk scores, so each of these distributions represent the risk scores within the respective groups of cases and controls. You can see that the increased shift for risk is what you'd expect for the cases. And this is what we calculated from the OncoArray. And then from the EpiCap study, you also see that similar shape also unfold. And then for the actual mean and the standard deviation for the polygenic risk scores for both studies were very similar, given the sample size that we had for EpiCap. And when we looked at this for the area under the curve, we also saw similar estimates of what we had from the OncoArray study. So here I'm showing the area under the curve plot, and in the black line here is just family history. So you have the null line, and then you have if you just included family history in the model. If you then included family history and the polygenic risk score, and if you also in red and then in green is if you just included the polygenic risk score. So you can see they're somewhat similar there. And these are similar to the estimates that we had from the OncoArray. But we really want to kind of demonstrate a little bit more of the utility of the polygenic risk score for prostate cancer. And we wanted to look a little bit closer at some of those substrata if we could. So we needed a large respective sample that would have the amount of information that would be needed. So this is where, at this point, we were able to extend to the UK Biobank. And if you haven't heard of the UK Biobank, this is just a brief description of it. I would highly recommend to go look this up, because for most people's traits, they're gonna have a large amount of information that could be available, particularly if they're aged between 40 to 69. And this is 500,000 individuals who have genome-wide association or GWAS data available for all these individuals, including on subsets of, I think, around 50,000. They also have exome sequencing and soon to be whole genome sequencing on 50,000. And there's a lot of informative information there. But similar, going back to the strategies and the stages of what you want to think about when you are developing your polygenic score, or if you're trying to validate it, is to make sure what you're validating in. And so for us, we really wanted to make sure we were validating just the polygenic risk score that we developed within our consortium, which was all, I'm sorry if I didn't mention this earlier, was all European based individuals. And so we wanted to make sure within the UK Biobank, we also did the same as well, because this was really just to point out of how well did that discovery set would work in this new validation set, this independent set. So here I'm just showing an ancestry plot of what the UK Biobank was and what it looks like, where you have Near East Asians and you have the individuals from the African continent and then the Europeans. And for over here, this is where we focused, was mostly within the Europeans. And then as we also moved through that quality control, we wanted to make sure that we were using the same type of case definition. So we weren't using individuals who had multiple diagnoses of cancers. They only could have prostate cancer as their primary diagnosis. So no secondary cancer diagnosis. And then within the controls, no cancer diagnosis whatsoever. So even if they had skin cancer or lung cancer, some others, we excluded them from the analysis at that point. And then we wanted to make sure we also excluded any related pairs as well. So when we kind of got to the final data set here, we ended up with over 7,000 prostate cancer cases and 192,000 controls. And you can see here what that distribution looks like for the polygenic risk score down here at the bottom. And so it doesn't look like it's actually significant, but it actually ends up being there. It's just the controls dominate. The other thing like we pointed out, you wanted to make sure you have everything on the same strand. If you don't have that, then it's going to really affect taking those summary statistics from your discovery GWAS and applying it to the UK or to your validation set. So here we're just showing a plot where we looked at the odds ratio that we had from our own discovery on the x-axis compared to the odds ratio for effect for prostate cancer within the UK Biobank. And this isn't the only way to check this. I mean, you got to look at allele frequencies and check the specific alleles and other such things. But this is one way of just kind of getting the general consensus across 178 SNPs that were included in this at this point. And you can see we have pretty good agreement here. So then just looking at the decile effects, or not quite the decile effects, this is slightly a little bit different. We did a comparison from the control groups, was for those who range from 40% to the 60% were considered the control group. So almost the interquartile range. And we looked at that comparison from just the OncoArray. So we just excluded some of the other GWAS data that we had from our ELLIPSE consortium, only looking at the OncoArray because we have a very it's a consistently set array that was done in 46,000 cases. And we looked at it within the UK Biobank, and you can see that it's almost identical point estimates for the validation. The only difference is slightly is the confidence intervals, which you'd expect from the difference of 7,000 cases to 46,000. And then when we did the area under the curve for the UK Biobank, we came up very closely to what we saw within just the OncoArray. So here you're starting to see improvements of just family history of 0.77, and then the individual polygenic risk score rising up to 0.8, and then you can see the combined RUC under there. So in conclusion, we felt like we had a 2-sample series that were independent from our discovery sample, and that these validated where our polygenic risk score is. We saw that this is, now there's definitely some limitations. This is only within Europeans. The AUC is relatively starting to climb. It's improving. And there's been a number of articles that have come out on recommending how this should be implemented, at least within some screening paradigms, and particularly with under age of 55. So we did validate that within the UK Biobank. And that's something that several people have been talking about, developing some studies looking at those who have a family history and under or at age 50, performing giving their GWAS and giving their polygenic risk score and determining whether they should have an annual PSA test or if it should be done more frequently. So future directions, I just want to point out a few here. The first one is, this is definitely was limited to just the Europeans. And I know the next speaker is going to talk about the importance of multi-ethnic polygenic risk score models. And this is definitely something that we're doing. So we currently have nearly 30,000 African-American cases and 30,000 African-American controls that we're looking at a couple different things, developing one, a completely separate polygenic risk model, also looking at how the European one that we built in this paper, how that would generalize to other populations. And we have another a Latino GWAS that has about 5,000 cases and 5,000 controls that we're looking there. And then we're currently talking to a recent Japanese study out of Riken Cancer Institute in Japan, where they had about 14,000 prostate cancer cases and about 80,000 controls, seeing how our models would overlap with those. The other thing that we're working on is the fine mapping within some of these locus. It's not always easy to define that you have 175 SNPs because sometimes you have conditional responses within the same locus, or you have multiple independent loci within the same locus. So within that OncoArray of those 46,000 cases and 30,000 controls, we've had a recent publication of doing some fine mapping, and this was using some Bayesian credible sets. And now we're trying to incorporate some of those in expanding the polygenic risk score to incorporate the ones that were independent. And then lastly is just additional genome-wide discovery. And then this is a little bit specific to cancer. We're a lot about power. So we're currently redoing our discovery analysis where we've taken the UK Biobank and we're adding them to the discovery series along with some other studies that we've managed to, who wanted to join the collaboration. So we're extending our discovery. So hopefully we'll find some additional loci. The bad part is we've lost that independent validation, or we're about to. So just in summary, make sure you have a large GWAS sample that the discovery sample will come from to get your betas and your standard error estimates. You're going to need some sort of similar or an independent target sample, depending on what your question is. You got to make sure that the SNPs overlap across both samples. And if you don't have exact matches, there are sometimes ways to use proxies, but you just got to be very confident in whether you imputed those and how much of a proxy they truly are within your sample. And if you're starting to cross into different ethnic groups, that might not be the best strategy at that point. And then applying those genome-wide discovery betas to your individual samples to construct your PRS and then to evaluate it. And the last thing here, just before we end, I just wanted to say there's a couple other resources here. There's just a recent publication in Nature Protocols that talked about how PRSs should be reported more in a standardized fashion, and how that what we should qualify as being independent validation and some other things. And this was just published out of Paul Riley's group. And then the other one I was just going to mention, I know not everyone here is cancer oriented, but I co-host with NCI's partnership, a webinar series that occurs once a month. And if you just look up Google Seek Space and NCI, you'll come up with the agendas that are coming up. But in 2 weeks, Nilanjan Chatterjee, who's currently now at Johns Hopkins, used to be at NCI, is going to talk about polygenic risk scores, both the development, validation, and particular applications. So I can promise you it'll be different than my talk. We didn't share slides or anything like that. And then obviously this doesn't happen in a bubble. This is a team that this consortium was across the globe. And so just wanted to make sure that I shout out for them. So a few minutes here left for questions.
Great, Fred. Thank you so much. So I'm looking at the chat box. There's nothing in the chat box yet, but while we're waiting for questions, and I probably should bring up the participants to see if there's any hands. I'll ask you, so the 175 or so loci that you're currently using in the polygenic risk score, I'm assuming they're somewhat common variants or not like rare variants. But as you mentioned, like UK Biobank and other studies are now including whole exome sequencing data, whole genome sequencing data is coming down the bend. How's your group visioning incorporating some of these more rare variants in a polygenic risk score?
So I think it's a great question. And there's two things we thought about. One was including them just directly into the polygenic risk score and you taking their effects, because we now have the larger studies that we can look at some of those associations. The second one is, I didn't go into it, but under the future directions was looking at some G by G. So the idea here is to look at those who are carriers. So for example, BRCA2 carriers. Are there certain polygenic risk score features that may differ whether you're a carrier versus if you're not? And now with some of the sample sizes, like you were saying with UK Biobank and even within some of our consortiums, we've been trying to figure out ways to do a couple things. One, impute within these rare genes, and if that's even feasible. And then we've also been adding sequencing data as people have been funded to do some sequencing studies. So we're looking at it as a stratification as well as just adding it directly to the risk score.
And just to follow up, none of the loci you're using right now, or maybe you were those imputed?
Yeah, so I didn't get into all the details because there's just so much time here, and I'm sorry for that. We do impute, yes, it is, and that was part of the QC protocol that I didn't go a lot into, but it is something that you have to be very careful of, because typically you're not going to have, even in your validation set, the exact same SNPs that you did from the meta-GWAS. So you're going to have to use some sort of imputation. And a lot of times the meta-GWAS is actually used the imputation, and some of what they discovered was just the imputed SNPs. So you do got to watch when you get into that. And that's where I would get a little bit cautious if you're starting to go into different ancestral groups or going into different arrays, that you're getting the same exact SNP. So I would really caution everyone to be very prudent about the quality control part. It seems like the easiest step, but it really shouldn't be. It needs to be something that you really focus on.
And we have a question from Dr. Farron Briggs. Farron, I don't know if you can unmute yourself, or do you want me to read it?
Yeah.
Okay, I'm going to read it. Okay, so the PRS and the assessments of heritability/liability are for joint additive effects. You mentioned epistasis as a future direction. How do you foresee that research going?
So right now I would say this has been an uphill run, and with like a 50-pound gorilla or a 100-pound gorilla on your back, because we are not getting anywhere with it. We've tried to look at it, and even with numbers that we're seeing, we're just not finding anything that we feel comfortable with to include within that model. Now, the one thing that we have thought about is the phenotyping sometimes for prostate cancer is the definitions can be very broad, right? If you've had prostate cancer, and if you're familiar with it, most men will develop it if they live long enough. So that's not the same as someone who gets it under the age of 55, right? If you got an 80-year-old who's developing it, they're going to be probably different diseases. So what we've been trying to think about is if we can just come up with some, maybe define the phenotype a little bit better clinically, and then we might be able to have better luck at looking at the G by E by removing out some of that heterogeneity. But the numbers then become an issue, because you cut down to 55, then you're losing a good part of your sample. So it's a balance, but yeah.
Yeah, we understand.
Yeah.
Great. Well, if there are no other questions, I don't see anything else in the chat box. I want to thank you again, Fred, for the great talk, and we will move on to our second talk. This one is will be given by Dr. Carla Marquez Luna. Dr. Marquez Luna received her PhD in biostatistics from Harvard University, and she's currently a postdoctoral fellow at Mount Sinai in the laboratory of Dr. Rondeau.
Hi, thank you, Dr. Crawford, for the invitation, and thank you to the organizers for inviting me to give this talk. And so I'm going to talk about the application of multi-ethnic polygenic risk scores to improve risk prediction in diverse population. I think Fred already did a very nice job in introducing the concept of polygenic risk scores, so I'm just going to go do a quick overview if there's anyone who recently joined. I want to focus this talk primarily in practical aspects of when you're trying to apply risk prediction in non-European populations. So polygenic risk prediction is, of course, it's a widely investigated topic because of its potential clinical application. It also helps us to have a better understanding of the genetic architecture of complex traits. And some important things to keep in mind is that it can achieve substantial accuracy at large sample sizes. And polygenic prediction outperforms the methods that use only genome-wide significant SNPs. So after the 10 years of the GWAS era, more than 20,000 novel associations have been characterized across many different diseases. And even the largest study analyzed so far has more than 1 million variants. However, making the promise of risk prediction more reachable nowadays. However, this is still a problem for non-European populations because most of these studies have been focused on European populations. As we can see, as reported in these two papers recently, by 2019, more than 50% of GWAS participants were recruited in the US and the UK, and the ancestry composition of these studied populations is primarily European. So we are interested in identifying potential ways to mitigate these natural biases that have already occurred with the current data that we have available. So I'm just going to give a brief overview of PRS methods, especially the simplest one. I don't think I need to invest too much time on that. And then I will move into talking about multi-ethnic polygenic risk score methods and some results that I have to share. So methods to compute polygenic risk prediction can be divided into two categories: the ones that analyze summary statistics only, and the ones that analyze individual level raw genotypes and phenotypes. In principle, methods that only analyze raw genotypes and phenotypes, such as BLOB and its extensions, can have a higher prediction accuracy. But what's the main limitation of this? Here, what happens is that we need to have available large data sets with genotype data that, as probably you will already know, this is very difficult to have because it's difficult to share across institutions, universities, etc. So we are particularly interested in methods that use summary statistics only. What this means is that usually when a research is published, a GWAS study is published, it is made available or easily available the effect sizes from the genome-wide scan. And as Fred mentioned, we need to have the direction of the effect. The allele that we are using as a reference, this is very important when we are trying to use data from other studies. We want the allele frequency and the sample size. And we also can take into account the correlation between variants called LD. And this can be inferred using publicly available data like the one provided in the 1,000 genomes. So we will focus on summary statistics methods only. And when we define a polygenic risk score, this is defined as the sum of effects of genotypes at a large number of SNPs using a SNP effect sizes estimated from a large sample. And it's an additive sum. It boils down to a problem of variable selection. That this means which SNPs are we going to include in our score. and also a regularization problem, which means about the estimation of the effect sizes. This means this is the data that I'm putting here in this equation. The most commonly used method is called pruning plus thresholding. This method is, applies a p-value threshold and optimizes over this value to balance a signal to noise ratio. So we are trying to find a way to include undiscovered causal variants by allowing SNPs in our score that do not necessarily reach genome-wide significance. And LD clumping, which means that we are removing correlated variants. And for any two correlated variants, we will keep the one that has the lower p-value. Typically, when we apply these methods to European populations, it is recommended that LD clumping parameter, for example, is around 0.1 or 0.2. So it's a stringent LD clumping threshold. When we want to apply a prediction in non-European populations, we have two options. So usually people will do a single ancestry method. That is, you will take the GWAS results from a study. Usually the training data will come from European samples. And the advantage from this is that we have European samples at large samples, from large cohorts. However, it's been widely reported that there's a relative decrease in prediction accuracy when these methods, when this GWAS training data is applied to non-European populations. For example, in the Hamilton et al. 2015, we observed a relative decrease in prediction accuracy that goes from 53% up to 89% in schizophrenia risk prediction in Japanese and African American populations compared to Europeans. The other option that we would have is to use training data from the target population if we have that available. If it is the case, it's likely to be available in a small to medium sample size. And the advantage of this is that we have this training data has similar LD patterns. What I'm going to talk about right now is about the advantages that we have when we optimally combine these two training data sets to build a single polygenic risk score. So just to delve into why we are seeing this relative decrease when applying European training data and predicting in non-European training data, some of the potential biases that would come up is one, heterogeneity in allele frequency. So there are undiscovered associated variants that are common in non-European populations, but are rare in European populations. Therefore, they are not included in the score. Another option could be heterogeneity in effect sizes, although recent studies might prove that this is not necessarily the case. And the other option that we consider more here is differential tagging due to differences in LD patterns. This means that because of the different different structural correlation between SNPs in different populations can lead to an error in assignment of appropriate risk scores. So if we, next. So just to illustrate this example a little bit more, for example, here we have these two, population 1 and population 2. I really like this figure because we have a causal variant, which is the red diamond that you can see here. And in this case, from population 1, the variant that tags the causal variant is D, but in population 2, it will be D. If we were going to use a GWAS estimated from population 1, and we use a method such as pruning plus thresholding to estimate the polygenic risk score and predict in population 2, it is likely that we will keep the variant D, but this variant is not informative when we predict in population 2. So, okay, what is the best way to compute polygenic risk scores? As I said previously, and this is just, we propose that when we combine different sources of training data, we can and we find a way to optimally combine them, we can achieve higher prediction accuracy compared to single ancestry methods. combining the different advantages that these different sources of training data provide. One is large sample size from the European training, and same LD patterns from the target population training data. So now I'm going to describe actually what multi-ethnic TRS will look like. I made this diagram just to try to to partition all the different options that you have when trying to do polygenic risk prediction in a non-European. So I already discussed this part, which is the one training. And then what would happen, oh no, so this is when you have one training population, you have these two options, European or the target ancestry. And the polygenic risk score would look like this. Another option is that you include an ancestry predictor. And then when you have two training populations, you have two options. The first one is you have the same data set target population that you use as training and test. This is the case when you do not have available an independent population. So let's say that you're interested, you have your own data set, you have your own cohort, and I don't know, it's a medium, maybe you have like 10,000 samples or 20,000. So you could split this into training and test and apply tenfold cross-validation procedures to estimate the GWAS summary statistics and then predict within your own data set. And if it is the case that you have access to an independent cohort, I don't know, maybe you have the UK Biobank data and you have your own cohort, and from the UK Biobank data you use the African samples, for example, maybe you could use these to get the summary statistics from those data, although I think there's like 7,000, maybe it's like small to medium sample size, depending on the trait that you're interested. But that will be the case where you have like an independent training data from that is non-European, and you have your own cohort that you wish to predict. So your polygenic risk score will look like this. We propose that you infer the mixing weights alpha 1 and alpha 2 using a linear regression to find the optimal combination. Typically, what people will do is to actually use a meta-analysis. However, the meta-analysis weights each PRS based on the sample size. But as I'm going to show in the results, this is not necessarily the optimal solution. And when the way we assess the methods is using adjusted R-squared, and as I mentioned, depending on the different training data sets that you have, either you have one and you're using that as testing and training, or you have one independent training and you're testing, you will use different layers of tenfold cross-validation to do, but I'm not going to get into the nitty-gritty of these results, but I'm happy to share the details for that. So some practical considerations that I wanted to highlight when applying these methods is that what we have seen is that even though when you apply PRS methods from Europeans to Europeans, for example, the R-squared LD for LD pruning is recommended to be very stringent, but what we have seen is that for non-Europeans, it could go higher to 0.8 or even 1. Of course, you want to match the ancestry from the LD reference panel to the ancestry from the training summary statistics. And so far, there are a wide variety of methods that we have available. But some of them might be complicated when applying to non-European populations, especially admixed populations, because of the complexities of admixture LD. So in our analysis in our paper that we published in 2017, we applied this method in simulations where we had Latino data and we had European training data from Wellcome Trust, and the target population in this case was the stigma type 2 diabetes genotypes that we had from, this is a Mexican cohort for type 2 diabetes. And we use tenfold cross-validation for estimating the GWAS sample sizes. So we use, in this case, we use both the target data was both our training and our testing. And here you can see that in general, European, if we, in single ancestry methods, the European outperformed the Latino, only 23% better on average, despite having twice as much sample size. And European plus Latino attained a 64 to 101% relative improvement compared to European and Latino respectively. So, and we see this across the board, but in the training sample size. In applications to real traits, we had an application of predicting type 2 diabetes in Latino data using the Sigma type 2 diabetes cohort. As training, we use Diagram, which is a type 2 diabetes meta-analysis that has more than 40,000 samples. And the Latino has around 8,000 samples. So as you can see, we have 5 times more samples in Europeans. What we can see here is that the European plus Latino outperform single ancestry approaches and standard meta-analysis. In particular, we see like a very large improvement in European plus Latino compared to single ancestry methods. And because I wanted to focus or highlight the practical considerations, as you can see here, the R-squared LD for European, the optimal R-squared LD for European was 0.8, and the p-value threshold was 0.5 on average, the optimal p-value threshold. And when doing the Latino, we see a very lenient p-value threshold, so we're including more variants, even undiscovered variants, and also the R-squared LD was very lenient, so we're including like very highly correlated variants. So another thing that we noticed in this example was that including an ancestry predictor improved prediction in the Latino population. So this is like the first PC. And we noticed that when we did, so in the single ancestry, Latino and Latino plus ancestry, we see this large improvement. But then when we applied the European plus Latino, we observed at the end the prediction accuracy stayed very similar, and this was because the European PRS was capturing a lot of the ancestry. So it was highly correlated with the PC. So this was an interesting result. Another thing that I wanted to highlight from these results is if you look at these weights. So I mentioned before that the typical approach that you will do is to use a meta-analysis. But what happens if you use a meta-analysis? You're weighting each PRS based on the sample size. We saw before that the European sample size was 5 times larger than the Latino sample size. So if we do a meta-analysis, we are going to downweight a lot the Latino PRS. However, when using a fold cross-validation scheme to estimate the mixing weights, we see that it is actually very similar, the mixing weight for the Latino, like to the European. So this provides a little bit more intuition about why it's important to optimize these mixing weights. We apply this method to other traits and populations, in particular type 2 diabetes in South Asian. In this case, we had independent European training and independent South Asian training from SAT2D cohort. And we predicted in the UK Biobank, at this time we had around 9,000, an effective of 900,000. This was, I think it was the first interim of the UK Biobank. And still we did see a large improvement of European plus South Asian PRS compared to the single ancestry methods. And we saw a 44% relative improvement compared to the meta-analysis. Again, you can see the R-squared LD, the optimal R-squared LD was very high compared to what you would see when using European and European. And the p-value threshold, for example, in this case for the European was very stringent, it was 10 to the -3. And the optimal p-value threshold for the South Asian was actually very low, 0.8. So basically we include most of the variants here. And the last application that I have is in Africans. And in this case, we also had independent training for the African sample, which is from Dai et al. We had around 20,000. This prediction is, instead of type 2 diabetes, it was predicting height. And we predicted in the UK Biobank, which at that time we had around less than 2,000 samples. And again, we see the same results. We see about a 30% relative improvement when using European plus Africans. There's a mistake there. Compared to meta-analysis and to the single ancestry approaches. In this case, the meta-analysis performed kind of similar, but still it was better to optimize the weights. So I'm running out of time. I just wanted to mention very briefly a recent application that I did with the UK Biobank, because this previous result that I showed was from my 2017 paper. And I recently applied this method, but this was using only European training and predicting in non-European. And I used it for training from 408K UK Biobank British individuals, and I predicted in South Asians and Africans. I use around 6 million variants, and I'm reporting results across 21 traits. And what I wanted to show just briefly was the comparison across many different methods that have been published recently, including one method that I'm about to publish that incorporates functional information. So we compared how multiple methods perform, especially some of them recently published, when predicting using European training and predicting in non-European. So in South Asians, we can see pruning plus thresholding, which is the method that I described previously and which was used to compute the previous results. This doesn't perform as well as LD-PRED, which is a more novel method. And the best performing method is both S-base-R and LD-PRED-FUNCT. This was published last year, and this is the method I'm about to publish that incorporates functional information. And this application was in South Asian, and we predicted in around 7,000 individuals. And then in Africans, we actually saw that using a functionally informed method predicted, achieve a higher prediction accuracy compared to other methods, in particular, pruning plus thresholding and LD-PRED. And this is interesting. We think that when you use like a method that incorporates many variants and function informed variants, you can gain some advantages even if you have only European training. So I want to thank the work that I presented here is the work that I did as a PhD student at the Department of Biostatistics in Harvard under the supervision of Dr. Alkes Price. I want to thank him and the people from his group, especially Per Rouleau and Stephen Gasol, and also my current boss, Ron Do, and thank you.
Oh, thank you very much. So I'm going to be monitoring the chat box here. If you have any questions, please type them in or attempt to raise your hand. In the meantime, I did want to ask for you to expand a little bit. I know you're running out of time, but when you said that you were incorporating function in your newly published method, could you define for us what you mean by function? I mean, is this like just exonic, or are you actually going to like do like EQTL stuff, or what were you doing?
So we use a method called stratified LD-score regression that incorporates about 56 functional annotations, and this is from like histone marks and like exon, like all these different categorical annotations. And also we incorporated continuous LD related annotations such as MAF allele frequency or recombination rate. So this is a method published by Finucane et al. 2016 Nature Genetics, and also Gassard et al. That was also developed by the group of Alkes Price. And basically we are giving a different weight to different SNP depending on how functionally enriched it is. So if we assume that the functionality of the genome is the same across different populations, which I think it's reasonable to assume, we think that this is why this method works very nicely when when we apply the method using even European training to other populations, because we're highlighting like the ones that are the most functionally informed. Yes.
Great. I see we do have a question for you here in the chat box. This is from Dr. Ellie Palmer. She's a postdoc here at Case. She said, great talk. Question, I noticed your note that LD pruning doesn't work due to admixture in the Latino-European assessment. How much does admixture influence the results and interpretation of multi-ethnic polygenic risk score studies?
Yes, so that note was about that I didn't use LD-PRED, which is a method that is very popular because of admixture LD. So admixture LD is basically this correlation that you see between SNPs that is not necessarily because of LD, but it's because of the different ancestry compositions in of ancestry in an admixed population, right? So it's like a spurious correlation. So this presents a difficulty when applying methods that incorporate LD, especially as a matrix, like continuously that model LD in this way, because you can include like spurious correlations. But this is when you apply when you have a training, Latino training, and you apply in a Latino population.
Yeah, I actually also wanted to expand a little bit on the admixture issue. So I know speakers don't have as much time to go into detail of all their data sets, but the Latino population that you were using in your initial European plus Latino comparisons, what was the admixture composition, and how would it differ if you were to sample, you know, obviously Latino, Latina, and Hispanic, it's like an umbrella term that catches a lot of different groups, right? So like what if you sampled Afro-Caribbean that fell within, you know, or high percentage of some kind of African ancestry mixed in there?
Yes, of course. So in this particular sample, this was the sigma type 2 diabetes cohort, which is Mexican population. In particular, the Mexican population, I think was like, the ancestry composition is, it was about 80% European and 20% other, like indigenous or even like a small percentage of African. So in this particular example, that was the ancestry composition of these, and it was mostly Mexican population recruited in Mexico City and I think LA. So it was a meta-analysis of these two.
So we have another question here in the chat box. How does the method perform if a different non-European cohort was used rather than a target non-European was split and used?
Like if we were going to apply like African to predicting Latino or something, I think is that, I'm guessing that's not a different non-European to predicting another non-European, but it's,
yes.
Yeah. I think I mean, I haven't done the analysis. I think it would be interesting to see the results, and maybe based on like the similar, maybe there are some populations that are more similar than compared to the European, the target population. There might be some others that are better proxy than the European. So that this can prove to be an advantage. I haven't done the analysis, so I think it will depend in a case by case.
Okay. Well, thank you again very much for your talk. It was quite interesting, and we're gonna move on to our last talk of the day. And let's see, this is going to be. Let me get this up for you guys. This is Dr. Athena Starlard-Davenport. So she's an assistant professor of genetics, genomics, and informatics at the University of Tennessee Health Science Center in Memphis, Tennessee. I should note that she's also an American Society of Human Genetics scholar as of now. And unfortunately, she couldn't be here live with us today to give her talk. She has some teaching obligations, as many of us do. This semester has started, but she was kind enough to record her talk for us that I'd like to play for for you live. And please note, she is available for questions via email. She'll give her email address at the very end of her presentation. And so please send me a note if you cannot hear the presentation as I play it.
Hello, my name is Dr. Athena Starlard-Davenport. I'm an assistant professor in the Department of Genetics, Genomics, and Informatics at the University of Tennessee Health Science Center in Memphis. Today, I'll be talking to you about research that I've been doing entitled Combining Community Outreach and Breast Cancer Genetics to Investigate a Polygenic Risk Score for Black Women in the South. The research taking place at the University of Arkansas for Medical Sciences described in this presentation was supported by genetic technologies. Breast cancer is the most commonly diagnosed cancer among Black women in the United States. Black women are also more likely to die from breast cancer. Racial disparities among women of color, particularly among Black women, is evident. The Center for Disease Control reported that Black women have higher mortality rates compared to all other racial ethnic groups. As you can see in this line graph, Black women, represented by the red line, have the highest death rates compared to Hispanic, American Indian, Asian, and European American white women. And for the first time in history, breast cancer incidence in Black women is now equal to that of their white counterparts, suggesting that as more Black women are diagnosed with breast cancer, more Black women will die from the disease. Breast cancer rates are even more devastating for Black women who reside in the southern parts of the United States. No region in the United States has as many high disparity states clustered together as the South. Louisiana and Mississippi have the highest racial disparities in breast cancer mortality, and in both of those states, the excess death rate among Black women is more than 60%, according to the American Cancer Society. Alabama, Arkansas, Georgia, North Carolina, South Carolina, and Tennessee all have excess death rates over 40%. And the cause of these breast cancer disparities is unclear, but studies have hinted to social structures and lifestyle factors that may contribute to the increased breast cancer incidence and mortality rates in Black women. Tools to measure and predict breast cancer risk are critical to potentially reducing breast cancer disparities in Black women. So breast cancer risk assessment tools, or BCRAT for short, were developed by scientists at the National Cancer Institute to estimate a woman's risk of developing invasive breast cancer within the next 5 years and within her lifetime up to age 90. The BCRAT is based on a statistical model known as the Gale model, named after Dr. Mitchell Gale, who is a senior investigator in the biostatistics branch of the National Cancer Institute's Division of Cancer Epidemiology and Genetics. The BCRAT uses a woman's own personal information to estimate risk of developing invasive breast cancer over specific periods of time. And so to do that, the BCRAT, it considers demographic information such as race, ethnicity, age, and breast cancer risk factors that are known to influence estrogen production, such as menopausal status, body weight parity, age at menarche, age at the time of the birth of the first child, and number of past breast biopsies and first-degree and second-degree family history of breast cancer. And so women with a 5-year risk of 1.67% or higher are classified as being high risk. So the BCRAT model, it has been tested in large populations of white women and has been shown to provide accurate estimates of breast cancer risk. The model was also tested in Asian and Pacific Islander women, Black, African American women, and Hispanic women using data from the Women's Health Initiative. It performed well in most populations, but underestimated risk in African American women with previous breast biopsies. And modification of the Gale model or the BCRAT based on data of African American women from the Women's Contraceptive and Reproductive Experiences Study, also known as the CARE model, still also underestimated breast cancer risk in African American women. Thus, risk assessment tools that can better predict breast cancer risk in African American women are critical to reduce breast cancer incidence rates in this population. Several studies have shown that combining risk estimates based on single nucleotide polymorphism genotypes with risk estimates from conventional breast cancer risk prediction tools such as the BCRAT can improve individual risk estimates for certain ethnic populations, particularly for European American and Asian populations. And so a growing number of SNPs, if you will, that are associated with an increased risk of breast cancer have been identified through GWAS studies. Most of these SNPs were discovered among women of European descent. These SNPs, they tend to be quite different in character from the BRCA1 and BRCA2 breast cancer mutations. And so while the latter confer a large increase in individual risk, they are found in very low frequencies in the population. In contrast, SNPs confer only a small increase in risk, but are found in much higher frequencies in the population. And so while no SNP is informative on its own, a polygenic approach to genetic screening could potentially improve estimates of individual risk and to create the possibility of individualized screening strategies, particularly for African American women. However, most of studies have been conducted in women of European descent. Studies have also been done in other ethnic groups, like I mentioned, including Asians. Fortunately, larger studies have also made efforts to identify SNPs specifically associated with breast cancer risk in African American populations through the Women's Circle of Health Study, the Black Women's Health Study, Carolina Breast Cancer Study, as well as the Multi-Ethnic Cohort Study. And the development of the consortium, including the AMBER Consortium, has done a great deal of this work. Currently, the AMBER Consortium has a combined total of over 8,000 African American breast cancer cases and over 17,000 healthy controls, making this the largest study of breast cancer in African American women in the country. Now this leads me to my research question of whether or not SNP inclusion improves BCRAT for Black women. This led me to my research objective, which was to determine whether a genetic risk score based on a panel of 75 breast cancer associated SNPs could improve the BCRAT risk prediction estimates of individual breast cancer risk among Black women. To test my research objective, we used a breast cancer cohort consisting of Black women in the state of Arkansas. In the state of Arkansas, breast cancer incidence rates have dramatically decreased due to state-funded breast and cervical cancer screening and diagnostic services for eligible Arkansas women. However, mortality rates due to breast cancer among African American Arkansas women are amongst the highest in the nation, especially among women living in the central Arkansas and along the Mississippi Delta region. Participants in this study were selected from a cohort of women recruited to study breast cancer in Arkansas called the Arkansas Rural and Community Health Project, formerly known as the Spit for the Cure. Accrual began in 2007 and consists of over 5,900 women, particularly Black women, and over 19,000 white women aged 18 to 100 years. Participants were recruited from several venues. A further 240 unaffected Black women were also recruited for the study at a Susan G. Komen Race for the Cure event held in April of 2014 in Jackson, Mississippi. Briefly, during recruitment to the cohort, baseline information on medical history, demographic factors, body weight, height, reproductive history, menstrual history, and family history of breast cancer was obtained, and a 2 ml saliva specimen from the participant was collected in an Origin DNA self-collection kit. Self-reported race and ethnicity information was based on data obtained from questionnaires that were also completed at recruitment. Participants also reported if a first-degree or second-degree family relative participated in any of the recruitment events. And so for this study, participants under the age of 35 were excluded since the BCRAT uses a BCRAT algorithm that's limited to women age 35 and older. For this particular study, a total of 878 Black women living in Arkansas were included in this study. Study subjects included participants who had a previous diagnosis of breast cancer, which included 319, one age-matched unaffected control, which was 319 per breast cancer case, and the additional 240 unaffected controls recruited out of Jackson, Mississippi. So this study and written informed consent procedure was approved by the University of Arkansas for Medical Sciences Institutional Review Board. And by using the breast cancer risk assessment tool to estimate breast cancer risk in our cohort, we observed that the mean BCRAT 5-year risk of breast cancer was 1.8%, with a standard deviation of 1.1% for cases, and 1.2% with a standard deviation of 0.5% for controls. The mean lifetime risk of breast cancer was 10.4% for cases and 8% for controls. Genomic DNA was isolated from saliva samples of the 319 cases and 559 controls using the DNA Genotek's DNA extraction kit. Whole genome amplification was conducted in the UAMS, or University of Arkansas for Medical Sciences Genomics Core Facility, and genotyping of 75 breast cancer risk assessment alleles was performed using the Illumina Golden Gate genotyping assay, also at the UAMS Genomics Core Facility. Blinded duplicate samples were also included to assess the reproducibility of the genotypes. The 75 breast cancer associated SNP panel was selected based upon SNPs identified as being associated with breast cancer risk by large-scale GWAS studies and had previously been used to derive a validated polygenic risk score using a European American data set. Because many of the SNPs have subsequently been found to have odds ratios of a different magnitude or are directionally different for Black women, the polygenic risk score used in this study was based on the previously reported odds ratios for African Americans. Collectively, we analyzed 75 breast cancer associated SNPs in the study together with the per allele odds ratios and 95% calculated confidence intervals calculated for the study population and the published values for both African Americans and Caucasians. Initially, we determined whether the risk score would improve without adjusting for age. Our findings show that for the unadjusted 5-year risk estimates, the combined risk score did not improve the area under the receiver operative characteristic curve. Similarly, for lifetime risk, the combined score also did not improve the AUC. However, when we looked at the combined risk scores in terms of the odds ratio per adjusted standard deviation, or ARPA, where the log 5-year predicted risk was adjusted for age, we did confirm that the combined risk scores for both 5-year and lifetime risk gave greater discriminatory accuracy than the SNP-based risk score alone, and compared to the corresponding unadjusted BCRAT 5-year and lifetime risk scores. Furthermore, incorporation of the 75 SNP-based risk score improve the odds ratio per adjusted standard deviation estimate from 1.84 to 2.09 for the 5-year risk estimate and from 1.84 to 2.07 for lifetime risk estimate. In conclusion, we observed that the discriminatory accuracy of the breast cancer risk assessment model is improved for African American women in Arkansas with the inclusion of the 75 SNP breast cancer risk alleles. The findings from this study were published in 2018 in the journal PLOS ONE if you would like to read it and for further details. However, this study is quite important, but additional studies to include underrepresented Black women in the United States, particularly those that reside in the southern regions of the United States, are necessary to validate the clinical utility of SNPs and risk assessment models. In 2017, I established the STAR study, short for Sisters Taking a Stand for Breast Cancer Research. The STAR study is a Memphis-based community participatory research study to help us understand breast cancer in Black women by collecting information to explore the biological, environmental, and epidemiological causes for the difference in cancer incidence and mortality among Black women in the Memphis area. And at the same time, we educate the community about strategies for early detection and prevention of breast cancer at various community outreach events. Here is an event taking place on April 1st, 2017, where we obtained informed consent from participants at a community event to raise awareness about breast cancer. After participants obtained consent and completed a health questionnaire, we collected a saliva specimen from each of the participants. We are currently working to identify biological and environmental influences on breast cancer risk by sequencing DNA samples in our cohort. Thus far, we have consented close to 400 participants, of which approximately 100 patients had a prior breast cancer diagnosis. Many of our participants in our cohort are either overweight or obese and live in impoverished areas within the Memphis community. So why is this important? Why are we focusing on the city of Memphis for this research study? Well, one, Memphis, Tennessee is one of the poorest cities in the United States. More than 60% of the population is African American. There's a strong history and a long history of racism and social injustices. And 21% of the population lives below the poverty line, including 30% of individuals under the age of 18. And there's a significant amount of health disparities here in the city of Memphis, including in cancer. In fact, Black women in Memphis have one of the highest breast cancer death rates in the United States compared to 50 of the largest U.S. cities. So to better understand the breast cancer racial disparities here in Memphis, my research team and I, we conducted a retrospective analysis on 1,342 Black women and 2,185 white women with breast cancer treated at one of the local Memphis hospitals to better understand racial disparities in regards to breast cancer. We found that Black women displayed increased mortality risk and were more likely to be diagnosed at advanced stages of disease compared to white women. Furthermore, in our study, Black women with triple-negative breast cancer or tumors that do not express estrogen receptors, progesterone receptors, or HER2 receptors had the highest death rate at 26.7% compared to white women at 16.5%. So more information about this study can be found in our paper that was published back in 2017 in the journal Cancer Medicine entitled Racial Disparities in Survival Outcomes by Breast Tumor Subtype Among African American Women in Memphis, Tennessee. In addition to these research studies, my team and I are continuing to explore genetic and environmental factors such as obesity that might contribute to racial disparities in breast cancer survival outcomes in underrepresented minority women in the Memphis, Tennessee area. In addition, we are wanting to combine our cohort with other cohorts to determine whether or not a genetic risk score based upon these breast cancer associated SNPs, including the 75 SNP panel that have been identified in Black women, could further improve risk prediction estimates of individualized breast cancer risk for Black women in Memphis and in the United States. I would like to acknowledge individuals listed on this slide at the University of Tennessee Health Science Center and West Cancer Center, the University of Arkansas for Medical Sciences, particularly Dr. Rhonda Henry Tillman, various volunteers and individuals that were very critical for community outreach efforts, including the participants, as well as funding support. If you have any questions about this research project or would like to contact me, I have provided my contact information here. Thank you. Have a great day.
So as I mentioned, Dr. Starlard-Davenport could not be with us today. She sends her regrets, but if you would like to reach out to her, we will have this posted along with her email address, and you can reach out to her directly for any questions that you might have. I want to thank our three speakers for today, Dr. Fred Schumacher, Dr. Carla Marquez Luna, and Dr. Athena Starlard-Davenport for great talks and a great first day of the North Coast Conference on Precision Medicine. Thanks everybody for coming, and I hope to see you again tomorrow at 1 o'clock. Have a good afternoon.